Dear participants, good morning to all. Welcome to this day 3 session on novel rules for non-muscle myosin 2 isoforms in renal epithelial transport and kidney function. Today, we have an eminent researchers with us, namely Dr. Indra Chandrasekar from United States of America. Madam is currently working as Assistant Scientist, Enabling Technologies Group, Sanford Research. Apart from this, she is also serving as Assistant Professor, Department of Pediatrics, University of South Dakota, Sanford School of Medicine, United States of America. The research areas of interest of Dr. Indra Chandrasekar includes understanding cellular transport pathways from an organismal and physiological perspective, developing novel tools and techniques to address mechanisms involved in specialized cellular transport, membrane remodeling events, and cytoskeletal dynamics in cells. Madam also working as co-director, Sanford Imaging Corps, United States of America. Dr. Indra obtained her bachelor's degree from Bharatiyar University, Coimbatore, India, with biochemistry as major and chemistry and zoology as minors. She received her MS degree from BITS Pilani, Rajasthan, India, with specialization of medical laboratory technology. She pursued her doctoral studies in biological sciences from International Graduate College Technical University of Brunswick, Germany. Later, Madam did her postdoctoral trainings from 2005 to 2013 at Department of Anatomy and Physiology and Department of Anatomy and Neurobiology, Washington School of Medicine, United States of America. Coming to the awards and fellowships, she received the fellowship sponsored by Ministry of Science and Technology. Also, she got FEBS Youth Travel Fellowship to attend EMBO or FEBS work at Austria 2003. She received Best Outgoing Student Award from Bangalore Jenny Private Limited during her Masters in the year 2000. Dr. Indra is an active professional member in various societies like American Society for Cell Biology, American Society for Nephrology, American Society for Biochemistry and Molecular Biology, American Physiological Society, etc. Madam is also the reviewer for the reputed journals like International Journal of Molecular Sciences and Cells. Dr. Indra Chandrasekar delivered many lectures and presentations as an invited speaker and keynote speaker at different international symposia, conferences, and meetings in different countries. She also has more than 21 research publications in reputed international journals like FASEB Journal, Molecular Biology of the Cell, Biophysical Journal, Cancer Research, Physiological Research, Journal of Cell Sciences, Traffic, etc. Madam, we are extremely fortunate to have you with us today in this FDP program, and I request you to enlighten all of us with your rich expertise in the field of molecular cell biology. I hand out the session to you, Madam. Thank you.
Yep, thank you. Dr. Erva, thank you very much. Thank you very much for your invitation to for asking me to present here to this group. Thank you for that nice introduction. That was not necessary, but thank you very much. I'm really happy to share with all of you what we are doing in the lab. I'm a cell biologist by training, and I started my lab in 2014. It's been 6 years of research in my laboratory. Although the title says that I'll be discussing the role of non-muscle myosin 2 isoforms in renal epithelial transport and kidney function today, I think for this group of people, because it's you guys deal with technology, it'll be interesting for you to understand biosensor work that we have done in the lab as well. The outline for my talk is I will introduce the research interests in my lab. Like I said, I'll be discussing 2 different projects in my lab. One is generation and characterization of this renal epithelium specific conditional knockout mouse model. Another one is a novel biosensor that we made in the lab to measure intracellular forces. I will give some time for questions and future directions that I will discuss as well. I'll be happy to take any questions if you want to ask me and interrupt me in between. But because it's a webinar, it's going to be difficult because I don't really see all of your chat messages, but you're welcome to ask me to go back to a slide if you want me to explain that again. So my laboratory interests are, like Dr. Erva discussed, understanding actin cytoskeleton associated proteins and their function from an organismal and physiological perspective. In order to do that, we had to develop novel tools and techniques and different model systems to study it. I'm not doing this alone. I am lucky enough to have team members that are very proficient and very good in what they do. So I have a postdoc, Dr. Carla Otterpole, who has a PhD in mouse genetics, and she has really took the mouse model project in the lab and accomplished a lot. The data I'm presenting today has been submitted to a really good peer-reviewed journal, and it's under revision, and we are hoping to get it published in the next 2 months. Mr. Brooke Basilman just joined the lab last year as a graduate student, and he is involved in the kidney project as well. So most of the Western blot analysis I'll be showing you today, he has done them. Mr. Ryan Hart was a laboratory technician in my lab for 3 years. He's currently a graduate student in UC Davis, and he single-handedly handled the biosensor project. He's a very good scientist and was really important for getting that biosensor project up and running, and that paper is also under revision currently. And of course, I can't thank the summer students enough. Every summer we have really good programs that brings in very good summer students to our lab. And I really enjoy training undergraduate students in exposing them to how scientific research in the lab is done. And, you know, I'm very hands-on. I go into the lab to train them. For me, the lab is my second family, so we really enjoy being in the lab, working together, and accomplishing novel things, trying to understand how the cell works. Now, moving on to science, I've been studying the actin cytoskeleton almost all of my career. My master's program through the DLPD programs division in BITS Pilani, I did it in Vision Research Foundation in India, and last year of that program is actually research. We do a year of research at my project to expose me to actin cytoskeleton. And I was fascinated by the eukaryotic cell and how cells function, and I did not let that passion go. So I applied to PhD programs in Germany and in France, but I got into a really good program in Germany, Technical University of Braunschweig, and that's where I went because there are really good old school cell biologists in Germany and France who are who can teach you fundamental cell biology, and I really enjoyed working there as a graduate student. Learned a lot from my graduate mentor, Professor Dr. Brigitte M. Jokusch. She was a well-renowned cell biology. Most of my cell biology knowledge, I credit her for teaching me the fundamentals and basics. So the eukaryotic cell is full of exciting biology. It's the fundamental functional unit. If you're a biologist, you have to understand the cell. And in particular, my interest has been in the actin cytoskeleton. The cytoskeleton of a eukaryotic cell is made of 3 main filamentous network. Microfilaments are the actin cytoskeleton, and it's named dependent on the size. The microfilaments are the actin cytoskeleton. Intermediate filaments, there are 5 classes of intermediate filaments and so many proteins in the intermediate filaments, and microtubules are the biggest of the filaments, or by size. The protein group that we are interested in is called myosins. It's a huge superfamily. I can spend one whole lecture discussing myosins, and I wouldn't be finished discussing all of it. One thing I would say is that all myosins bind to actin, and they are all ATPases. They are enzymes. They generate energy by hydrolyzing ATP. By generating that energy, they can slide or walk on actin. So they are all motor proteins. The many functions for myosin proteins are in cargo and organelle transport. It's really important for maintenance of cell structure, especially actin-based structure, because it gives the cytoskeletal strength. It's very crucial for cell division and also for muscle contraction. So as you can see, for so many cellular processes, for life sustenance, myosins are important. There are about 20 plus classes of myosins across eukaryotes. We are still discovering new myosins. What we study in the lab is class 2 myosin, or non-muscle myosin 2, which is kind of a misnomer because it's ubiquitously expressed. It should have been named cytoplasmic myosin. The other class 2 myosins in mammalian system are cardiac myosin, smooth muscle myosin, and we all know that this class 2 myosin in heart muscle and smooth muscle and skeletal muscle is really important for muscle contraction. This is the unit that, along with actin cytoskeleton, brings about muscle contraction. What we study in the lab is non-muscle myosin 2 or cytoplasmic myosin 2, which is ubiquitously expressed in most cell types. I'll move on to the biosensor project. This came about because we were curious about, can we measure the force generated? We talk about myosin motors all the time, and myosin 2 especially, I told you they form bipolar filaments. I'll discuss this a little bit more in the next slide. And can we really measure this force? Was just a curious question. Because when we say that it's really important for the force generated along the actin cytoskeleton was really important for vesicle fission during the early process of endocytosis, my question was, do we know how much force is needed for that? So that's what brought this along. And I've not talked about myosin 2 structure earlier. So non-muscle myosin 2 is a very interesting protein. It's a hexameric protein. It has 2 heavy chains, the globular head domain and this coil domain. The MYH9 and MYH10 genes actually code for what is the heavy chain. And there are 4 light chains that come from completely different genes, and they meet in the cytoplasm, probably to get translated or transcribed together. And they make this unit with the 2, the hexameric protein. And the myosin light chain gets phosphorylated, and that activates. It's actually an inactive molecule due to this intramolecular interaction, and the phosphorylation releases the intramolecular interaction, and thereby it will enable it to form this bipolar filament. There is a filament assembly domain at the end, and they can form the bipolar filaments that lead to contraction. Sweta, you can ask your question if you have a question. Or we can wait until the end. I'm fine with that. So what you're seeing here is a very nice movie of a 2D cell cultured, which is expressing a GFP myosin 2B, GFP fusion protein of myosin 2B. And a small polypeptide that was discovered 10 years ago that has affinity for actin cytoskeleton. It's called Ruby LifeAct. And we can image it with two lasers. And as you can see, they form very nice bipolar filaments, and see the contraction that's happening in those filaments. And these cells are plated on fibronectin, if I remember correctly. That's why they are forming these really long stress fibers. We call them stress fibers. So what we wanted to do was to expand the toolbox for measuring force, tension within, measuring tension and force within the cells. 10 years ago, a 2010 paper described a very nice fluorescent resonance energy transfer based tension sensor module. So what they did in this paper was to discover a natural spring molecule, naturally occurring spring from a spider silk protein, I think. And this spring has inherent mechanical, it's like a tensile strength. So it's spring. If you put this on a protein that's experiencing force or sensing force, depending on, and you put a donor fluorophore and acceptor fluorophore on both ends, because it's fluorescence resonance energy transfer. So in here they put donor fluorophore that's going to sense blue light, and it will transfer the energy to the acceptor fluorophore. When you put this whole tension sensor module, the FRET-based tension sensor module, in a protein that's experiencing force, if it is in a relaxed state, these two donor and acceptor molecules are close to each other. The spring is in the relaxed state, so there is high FRET. There's very high levels of fluorescence resonance energy transfer, as opposed to when you have a tense state, when this molecule is under tension, you have low FRET. So what we did was we designed a non-muscle myosin 2B. This is MYH9, a non-muscle myosin 2B with this spring inside. So we cloned elegantly into a really good area of myosin 2 molecule, this tension sensor module. And we now believe that this molecule, myosin 2 molecule, can sense and report the tension or force generated or sensed by myosin molecules in live cells. And like I said, Ryan was the one who did the work. So this is Ryan with Jing and me in one of the cell biology meetings that we were presenting the work. In order to say that if this protein is actually functional, because we just introduced this huge spring into the myosin molecule, we had to prove that when we express it in cell types that lacks the endogenous protein, they do form filaments. And when we proved that in the absence of, in the myosin 2B null mouse embryonic fibroblast, which do not express endogenous myosin 2B, when you express myosin 2B TSMOD, they do form filaments, and those filaments co-localize. You can stain for it using a myosin 2B antibody, and they co-localize with actin cytoskeleton as well. So they are good. And this is a collaborative project between, I am the biologist who designed the molecule, but then I'm not the technology person who does the FRET experiment. For that, we collaborated with Dr. Jing Liu. He was in South Dakota School of Mines, where Divya Kota was a graduate student at, but he moved to Indiana University a couple of years ago, but we continued this collaboration, and Divya was a student who continued to work on that. So what they did was to do the fluorescence lifetime imaging using the special microscope, which detects, there are a couple of ways to do the analysis for the fluorescent resonance energy transfer. You can see whether the acceptor is taking the energy in, or what you can do is when the photons are released from the donor molecule, you can look the energy, what is the fluorescence lifetime of the of the donor molecule. So these are images showing our FLIM-FRET studies. The top panel is a fluorescence resonance energy, fluorescent lifetime imaging image, and the corresponding FRET image. So if we made the second panel is showing the myosin 2B values, and the first one is actually our donor-only control, where we have to expect high lifetimes and low FRET, low transfer, but high lifetimes, because there is nothing else to transfer the energy to, because it's only donor molecule that's attached to the myosin 2B. And then in the other control, instead of the spring, we replace the spring with 5 amino acid linker. What this does is it's the closest proximity that the donor, the fluorophores can be at, because there is no spring there. So we expect a very high FRET level in this particular control. We also made a dark acceptor control, where we made a point mutation in the acceptor fluorophore that renders it non-functional, so it cannot take any energy. So this one has to give us a really low FRET again, because there is no energy transfer. So consistent with the design of the control molecules, the donor-only control had very low FRET percentage. Similarly, the point mutation which renders the acceptor fluorophore non-functional also had very low FRET percentile. The 5AA replacement control actually had the highest FRET value that our FLIM microscope recorded. In the myosin 2B TS-mod, on the other hand, had a very dynamic range of FRET percentage that we could record using it, which was within the limit of the mechanical sensitivity of the tension sensor module itself. So this is a movie, a FLIM movie that Divya made. This is a cell type that is stably expressing our TS-mod. The colors changing actually indicates the force changes. So the lifetime continuously changes in different areas of the cell as the cell is moving forward. So we finally have a really good biosensor that is capable of detecting dynamic force changes in migrating cell. And this is the how we analyze the data. So these are time-lapse images of the 31-minute movie I just played. And, you know, based on the previous publication, we know that those lifetimes or the FRET can be converted to force, because there is a equation that they published based on the tensile, the mechanical sensitivity of the spring. And so we directly converted those FRET percentages to the force. And as you can see, this is a force map, and you can see changes in multiple different areas of cells, different forces. You also can now plot individual areas, region of interest, and see along so many microns of that particular fiber, how is the FRET efficiency changing? And that can it gives you a value there. And so it's a spatial distribution of FRET efficiency. And you can also get temporal FRET efficiency, meaning select a region of interest and then look at the FRET efficiency changes over time. In conclusion, we have generated a novel biosensor that's capable of reporting on intracellular force dynamics. We are the first to apply this particular FRET-based tension sensor module to a molecular motor protein. And applications for this can be in biomaterials bioengineering, cancer biology, or stem cell biology. Of course, when you generate a tool, you also have to be aware of the limitations of the tool. What we do not know is exactly what it is measuring. because like I said, myosin is ATPase, they generate force. Is it actually measuring the force generated by the filaments, or is it actually measuring the force experienced by the filaments, or both? We really don't understand that right now, and we are also limited by the mechanical sensitivity of the tension sensor module itself, the spring itself. I think right now, if you convert the foot percentages to the force, it's about the linear range is about 1 to 6 piconewton. So I don't think in all our cells we are only going to experience 1 to 6 piconewton. The forces are going to vary. So you have to design a new spring in order to measure different variety of forces that cell is experiencing. I don't do that kind of work. There are experts in the field who design that tension sensor module, and I leave it up to them. And there is new tension sensor module with a wide range of mechanical sensitivities available. We will be interested in applying that to myosin 2 proteins. And right now, the paper is actually on bioRxiv. Anyone can go and look at it. That's the data I presented. We are on the process of revising this manuscript and submitting it back again. And with that, I'm done. And I thank for all of you for listening. And I thank the people who are involved in it, and the funding and the resources at Stanford Research, and all my collaborators and advisors. Thank you very much. That is my contact info. I really enjoy hearing from people. And if you email me, and if I can help you in some way, I will be more than happy to help you. Or if I don't know what answer for what you're asking, I will ask some of my connection if they can help you. Thank you.
Thank you, Dr. The participants can post the questions in the chat box if they have any.
It's an interesting point. I don't know if the congenital kidney stone is related to the protein being not actively being transported. It is possible. It's definitely possible that the protein is getting stuck in the ER. But I think what I would say is it's not the cause or the reason why they are getting kidney stones. I think the reason for that would be, deregulated ion transport. So, like I said, if you're calcium oxalate or one of those ion transport in the thick ascending limb is deregulated because of mutations in neuromodeling, then yeah, that probably is what is causing kidney stones. Which method is used to measure urine sodium and potassium levels? We have a Siemens instrument, where you run your human blood samples. We have an instrument, and we buy kits where you run serum and urine samples. We have a Siemens blood analyzer. Interesting aspect. This is so expression of MOH9 and 10 is very interesting. So what I would say is, like I explained to you, this is a complicated molecule, right? It's a complicated protein because MOH9 and MOH10 only code for the heavy chain, and other genes code for these light chains, and what chaperones are involved, and how the transcription and translation of all these molecule genes are happening, and when they meet, and or is it actually a combined transcription translation that's working to put this protein together? It's really not very well understood. So, what transcription factors are really regulating MYH9 and 10? Or any other myosin molecule is not, from my understanding. I don't know the answer for it. And I think beyond transcription, how these different gene products get together to make a functional protein is, in my opinion, very interesting, and we don't know the details of it.
Yeah. Any other questions?
Hello, ma'am.
Yes,
Hello.
hello, hi.
Dr. Shweta here from MIT University, Rajasthan, Jaipur.
Okay,
Ma'am, it's
hi.
very nice presentation. Very glad to know about the kidney and different molecules in the kidney function, muscular function of renal. I would like to know, as you tell us about the biosensor part, that is very interesting. But could you please explain the role of biosensor in cancer biology?
Hmm. One thing I would actually say is, when you develop a tool, for example, I can only perceive so much, right? I developed the tool because I was genuinely curious. Can this tool even work? And can we measure force changes in live cells? Um, when I say maybe it will be useful for stem cell biology or cancer biology, from what I am thinking about, is if you actually understand force that should be generated within a normal cell, right? Now, whatever normal situation the cell is in, and if you have a cancer cell type where the forces are changing, because clearly in cancer cells, we not only have the cell division that's going crazy, for which myosin 2 is really important, and force is really important, and they are becoming highly migratory. So it's possible that if we can measure the force changes in the cancer cells, they might be different, and the way they react to chemotaxis or any other hormonal uh, signaling cue is going to be very different. But it's up to the cancer biologist or stem cell biologist to use the tool. I can only perceive the tool so much. So you can come up with an idea to use the tool in whatever way you want. I am happy to share the tool with anybody who wants the tool.
Okay, ma'am. Thank you very much, ma'am.
You're welcome.
And second, I would like to know the source of sting, which you have explained that it's obtained from the natural source. So that is present naturally in the cell. So which type of sting molecule is that?
So I did not develop the tool, but develop the spring. So like I said, it's a
Okay.
2010 paper in Nature. I think it's a collaboration between a really good cell biologist and engineers. They screened for naturally occurring spring type molecule, and they said this one is a spider silk protein, I think. Yeah,
Okay.
this
Okay.
the spring is actually from spider silk protein.
Okay. Ma'am, is it same which is used for biosteel preparation?
I really don't know. I don't know. You
Okay.
can
Okay.
look it up.
Thank you,
So
ma'am.
look up those original papers, because those are the question you're asking is a very engineering question, and from my understanding is, I know what the spring is from, but I really don't
Okay.
know.
Okay. Okay. Thank you, ma'am. Thank you very
Yeah.
much. Very nice
Thank you.
presentation it was.
Thank you very much. Thank you. Thanks for attending. And
Yeah. Welcome, ma'am.
is there a near-term possibilities for getting genomic medicine too for CKD patients? I think there is a lot of, and there was a question about, is there a difference in progression of end stage? And thank you very much for everyone who is saying good presentation. Thank you very much for listening to it. And I don't know if there is, I'm sure there is a difference in how the kidney disease progresses in mice and humans because, one, the kidney size is different. But most cases, it's not like, in my experience, if I understand kidney disease very well, I think kidney is an organ that you can go to any mammalian system and say it has to function, otherwise you're going to die, right? So I think in that way, it's very common. And as I showed you all a lot of data, the proteins that are expressed are mostly similar in mouse and humans. Is there any near-term possibilities of getting genomic medicine tool for CKD patients? I think people are already doing that. I don't know what you mean by genomic medicine, but I think like places like MedGenome is offering exome sequencing and then trying to understand what particular gene mutation is in certain patients with kidney disease. Yeah, that's happening. But as far as the cures, I don't think so. Thank you, Dr. Balamurgan.
Yeah, dear participants, if you have any questions, you can write to Indra.Chandrasekar at sanfordhealth.org, which is displaying on the page, the desktop page. This is the time to thank our speaker for today, that is Dr. Indra Chandrasekar. Madam, the content you have shared with all of us on biosensors and other molecular cell biology aspects on myosin 2 was very interesting, informative, and quite useful for many of us. We are really happy and benefited out of your talk. Thank you, madam, firstly accepting my invitation to deliver the talk as part of this 5-day faculty development program from webinar series on latest trends and future prospects of biotechnology being organized by National Institute of Technology, Andhra Pradesh. Thank you again for being with us today in this event and enlightening us with your rich research experience. Thank you very much.
Thank you. Thank you very much, Dr. Erba.
Thank you, madam.